Hello everyone, I'm Sidhvita, a member of Krino Forensics. In this video, we are going to discuss semen as a forensic evidence. Semen is one of the most crucial biological evidences in forensic science, especially in sexual offense investigations, because it provides both associative evidence and individual identification through DNA. So, in this lecture, we will understand what semen is, the structure of spermatozoa, the physical and chemical composition of semen, presumptive and confirmatory tests, and finally its forensic significance and challenges. So, now let's begin with the introduction. Semen is also known as seminal fluid, is an organic fluid secreted by male reproductive system. It primarily contains spermatozoa along with other substances that support sperm survival, motility, and transport. Semen is produced by gonads and accessory glands, which includes the seminal vesicles, prostate gland, and bulbourethral glands. Its main function is to act as a medium for transporting spermatozoa, making it essential for human reproduction. From a forensic point of view, semen is considered genetical material because sperm cells contain nuclear DNA, which is extremely valuable for identification. Because of this, semen collection and preservation are important in both medical and forensic context. Now, let us understand the morphology of spermatozoa, which is very important for microscopic identification. Human sperm cells are highly specialized and consist of four main parts. The head contains the nucleus, which is densely packed with chromatin. It is covered by acrosome, which is rich in enzymes that helps the sperm to penetrate the ovum. The neck contains centrioles, which play a role in cell division after fertilization. The midpiece is packed with mitochondria, which is responsible for ATP production. The tail is also called flagellum, provides motility to the sperm through lashing movements. This unique structure makes sperm cells easy to identify under a microscope. Now, let us move to the composition of semen, starting with its physical properties. Semen is usually white or transparent in color. It has a thick, sticky, and glary texture. It has a characteristic seminal odor. The specific gravity of semen is approximately 1.028. The pH ranges from 7.35 to 7.5, making it slightly alkaline. The average volume per ejaculation is about 3 milliliters. Now let us look at the chemical composition based on the glandular contribution. The testes and epididymis contribute about 5%, containing roughly 80 million spermatozoa. The seminal vesicle contribute the largest volume, around 50 to 65%, which provides energy and supports sperm function. The prostate gland contributes about 20 to 30% and secretes citric acid, zinc, acid phosphatase, etc., which help in semen liquefaction and sperm motility. The bulbourethral glands contribute trace amounts of mucus which lubricates the urethra. Apart from glands, certain supporting cells also play a role, such as Sertoli cells secrete fluid that nourishes and transports the sperm. The ductally efferentness modifies fluid composition through absorption. The principal cells of epididymis secrete glycerophosphocholine, which prevents premature capacitation of sperm. Now let us move to the presumptive test used to detect semen. The most commonly used test is the acid phosphatase test. This test is based on the high acid phosphatase activity found in semen. The reagents used are sodium alpha naphthyl phosphate and naphthanyl diazobluB. Here, a rapid purple color indicates a positive result. The next test is fluorescence test. The fluorescence test uses potassium iodine, and water. A positive result is indicated by the formation of a dark brown crystals. This test is usually positive within 24 hours of post intercourse. The third test is Barberio's test. The Barberio's test uses picric acid. A positive result shows yellow needle-shaped crystals of spermine picrate indicating the presence of spermine. The next test is thin layer chromatography, which can detect the choline and spermine even in stains up to 5 years old. We can also use alternative light sources which are used to visualize semen stains under UV or visible light, especially on different fabric colors. Now let us discuss confirmatory tests which provide definitive proof. The microscopic detection of sperm cells is considered the most reliable confirmation of semen. Another confirmatory method is crossover electrophoresis, which detects the P30 antigen, a semen specific protein. Here, the antigen from the stain and the anti P30 antibody are placed in wells. Formation of precipitin line confirms the presence of semen. Now, let us understand the forensic significance of semen. Semen helps in confirming sexual activity. It helps in linking the suspect to the victim or crime scene. It provides DNA for individual identification. It is crucial in rape and sexual assault investigations. It helps in determining ejaculation or type of assault. In some cases, it can also help estimate the time since intercourse. Now, despite its importance, semen analysis have several challenges. Semen samples may undergo degradation due to heat, moisture, and time. In cases of azoospermia, sperm cells may be absent, such as after vasectomy. Mixed stains with other body fluids complicate analysis. Presumptive tests may give false positives. Sometimes there is a low or degraded DNA yield. Chemical inhibitors may interfere with the testing. Washed or laundered evidence may be removed semen traces. These are also legal and ethical considerations, including consent and proper handling. Risk of contamination exists during collection or laboratory analysis. Finally, semen may be difficult to distinguish from similar body fluids. So, to conclude, semen is one of the most powerful biological evidence in forensic science. Despite certain challenges, proper collection, preservation, and analysis can provide critical information that supports justice. Thank you for your attention. I hope this lecture helped you to understand the forensic importance of semen. Please drop your comments below if you have any questions or want us to explain any forensic topic. We would be happy to cover them in future videos. And the presentation document will be shared on our social media channels. You can directly access the document from there. Stay tuned for next video to learn with Krino Forensics.